Transcription in prokaryotes. Transcription is the process of using DNA as a template to make a strand of RNA. This process starts when the RNA polymerase becomes a holoenzyme. In order to find and bind the promoter region, the RNA polymerase must be a holoenzyme, meaning it consists of its core subunits and sigma factor. The sigma factor is what finds and binds the -10 box and -35 consensus region of the promoter, which are upstream of the +1 site or transcription start site. The binding causes the DNA strand to separate in the polymerase and form a transcription bubble. The strand being transcribed is the template strand, which is the bottom strand. The RNA polymerase should be moving along this strand in the 3' to 5' direction, meaning the RNA strand being produced will be complementary and will be produced 5' to 3'. This strand will be identical to the top strand of DNA called the coding strand or non-template strand, except the mRNA strand will have uracil instead of thymine in it. The RNA polymerase catalyzes phosphodiester bond formation. These bonds are formed when the 3' OH group from the last ribonucleotide attacks the 5' phosphate group of the incoming ribonucleoside triphosphate. This process cleaves off 2 phosphate groups from the incoming ribonucleoside triphosphate as the nucleotide is added. Once about 9 to 12 nucleotides are formed joining the RNA strand, the sigma factor will dissociate and the RNA polymerase will move from the promoter region down the DNA, catalyzing the addition of more ribonucleoside triphosphates to the RNA strand. Elongation continues until a segment of inverted repeats is transcribed. These form stem loop or hairpins in the RNA sequence, which signals termination and causes the polymerase to pause. In row-independent termination, following the inverted repeats in the template is a poly-A tail, which is a string of adenine residues. The RNA polymerase will transcribe a poly-U tail. These A's and U's bind the DNA and RNA together weakly, which allows them to dissociate. As was seen, the transcription process started with the polymerase finding and binding the promoter region. If per se the -10 region was missing from the promoter, then the RNA polymerase would have trouble finding and transcribing the gene. If this gene encoded for an enzyme that metabolized sugar, then the prokaryote would no longer be able to metabolize sugar efficiently.